Thank you very much, Petra, for this wonderful introduction, and also all of you for the invitation to be here. One of the big challenges in chemistry and the big contributions of chemistry to society has certainly been and continues to be learning how we can convert one energy form into another energy form. How can we convert light into electrical stimuli? How can we convert chemical stimuli into molecular motion? We heard a few nice talks yesterday to that topic. So in order to improve our abilities to convert one form of energy into another, sometimes it is good to look into Mother Nature and ask what kind of inspirations can we get when studying biological molecules. And one of our key interests in my own research is to learn how we can convert mechanical stimuli into biochemical signals. And this is what I would like to focus on today. So this entire field of looking how molecules respond to mechanical stimuli was only made possible again by a few Nobel Prizes, showing that proteins have mechanical properties that are very different from polymers. And 3 papers appeared in the same month, all showing that you can pull single molecules, and particularly if you pull proteins, that proteins can be destroyed in a very nonlinear fashion and by a breakage of these domains one by one by one. So it's a very highly nonlinear response. In the meantime, certainly many proteins have been pulled. Many have been characterized mechanically. And the next big question is, are the forces that cells generate sufficiently large to mechanically deform a protein? The second big question is, does nature take advantage of pulling on proteins to switch important functions? And I would like to really explore both topics today. So if you go back, and probably you remember the day when the human genome was solved, and the big excitement that we will be able to really solve many, many diseases just by knowing and having information about the genome. And then essentially, huge next wave of exploration started to putting together sequence information, structure information, the information about functional states of proteins into these signaling networks that for everybody who's not from biology are always very confusing, very complex. Yet the goal is certainly to finally learn how we can go from a sequence to the function to predicting a cell phenotype, the behavior of a cell upon a particular stimulation. However, if you think, and if mechanical forces can distort the structure of a protein, then something that we can still not yet enter even structural databases might happen. If mechanical forces are now acting on a protein, you might change the structure, but if you change the structure, you're also likely to change the function of this protein. So perhaps by mechanical forces, you can just switch into a different cell phenotype. So there are no experimental techniques available so far to determine protein structures of mechanically stretched proteins at high resolution. So computational tools play a major role, but then there's also no way of entering that information into databases to make it available for many researchers to come. So shortly after these first AFM studies on Titan were published in a wonderful collaboration with Klaus Schulten that is still ongoing, we asked the question, what defines mechanical stability? And initially in the protein folding community, people described this energy landscape and all the local and global minima, and certainly the folding is really controlled by hydrophobic residues that try to protect themselves from water. So the first idea was certainly also that it's the same hydrophobic residues that prevent unfolding upon a mechanical stimulus. However, the first simulations of the published structure immersed in at that time, this was all we could simulate, bubble of water molecules revealed a different picture. And the different picture is that there are force-bearing hydrogen bonds, backbone hydrogen bonds. And they need to be strained, they need to be broken in order for a protein to unfold. And so sometimes the cluster of these hydrogen bonds is really the determinant, not how many hydrophobic residues might be buried in the rest of the protein. So this is 10 years ago. Where are we today? First of all, the first question is, where can we potentially find proteins that are mechanically stretched by cells? And therefore, we have to just look briefly into the events that might happen if a cell is in a medium, starts absorbing to a surface, to a material surface, to a biomaterial. The cell rapidly forms cell adhesion sites. The cytoskeletal reorganization is happening. Finally, there are then some stimuli that cause nuclear functions to be changed. And what the community has studied in detail in the last 20 years now are how cell adhesions are formed, how the cytoskeleton is reorganized, but we still do not know why a cell, for example, can recognize certain topographical features and how this information, the cues that come from the cell environment are finally translated in the nucleus into different gene expression profiles. So cells can sense physical properties in their environment, chemical composition through these adhesion sites, surface texture, perhaps the rigidity of materials. And so we need to look now a little more closer into these contacts that the cell forms with the environment. First of all, it's important to notice that the cell forms physically linked networks of proteins that closely attach to each other, and that you essentially connect the contractile cytoskeleton, where you have motors that pull on filaments, to the outside world through these junctions, which are called Integrand Junctions. So if the cell can apply mechanical force, all the proteins that are part of this force-bearing protein network have the potential to being stretched, and also the potential to being altered functionally. And what I would like to focus upon now are extracellular matrix proteins that define the integrity of connective tissue. And first of all, let me show you, if we would make a little stretch in our skin and look underneath, we would see fibroblasts that make our skin and are responsible for remodeling tissue, or they are responsible for wound healing. And if you now look into such a scaffold, what you might see are fibroblasts here in blue, and here you see a lot of green fibers. And now this was supposed to run. Let me try again. Can you please activate the video here? So what you see now is that the fibroblasts grab fibers, stretch the fibers, let go, grab, stretch, and let go. So indeed, they can apply sufficient forces to stretch a fiber, but does the stretching relate to functional changes? I would like to illustrate that the extracellular matrix has a lot of integrated mechanochemical nanoswitches. And the illustration here should be done with bacteria. And this is one of the most likely bacterial infections that you might get in a hospital, as aureus. You see the bacterial body here, then long appendices on the surface of the bacteria. And essentially, those are long peptide chains that can again recognize the protein fibronectin. And they do that in a very interesting way. Fibronectin has many structural domains here called type 1 domains, type 2 domains, type 3 domains. And these long peptide chains, they can bind not just one binding site, but several binding sites in sequence. So it's a multivalent contact that can be formed. What I would like to show you now, that if we stretch these fibers, or if cells stretch fibronectin fibers, you can destroy the binding epitope for S. aureus. And let me show you first what kind of assays need to be developed to even illustrate such a phenomenon. There were studies in the bioengineering community in the 90s where they took droplets of protein, they came in with a sharp tip and could pull protein fibers out of these droplets. Here what you see are fibronectin fibers that are deposited now on the substrate. And they try to utilize that to make scaffolds for neuroregeneration. We adopted the same technique, but now deposited these fibers on stretchable substrates. And then we took peptides from different bacteria for which we also had high-resolution structures and labeled them with a second color. And by doing so, we can, pixel by pixel on our fibers, determine the ratio of peptide to fibronectin that is being bound. And what we see very nicely here, if you stretch such a star of fibers in one direction, you essentially eliminate the binding of the bacterial peptide in the stretched fibers, whereas a lot of bacterial peptide can bind to the relaxed fibers. How does that work? What is the design of this switch? Again, we need computational techniques. The NMR structure was known. And what you see is, and if we could just go back to the beginning of the simulation, is an equilibrium structure. You see the green peptide here. You see the green bacterial peptide that forms backbone hydrogen bonds at first. And then as you stretch the underlying fibronectin template, the bacterial peptide pops off. And essentially what you've done was, initially you had two binding epitopes at a certain distance. And by stretching the template underneath, the bacterial peptide cannot bind any longer. We then investigated a lot of patients in get heparin treatments. Heparin binds to the same domains that are targeted by bacteria, where the heparin or enriched heparin levels in the blood would destroy mechanosensitivity. What you see is that heparin indeed competes for the bacterial binding sites, but the peptides, the bacterial peptides that bind, they still bind in a mechanosensitive manner. So why is this interesting? Maybe with such a small, simple sensors, bacteria could potentially discriminate between healthy and diseased tissue. Because imagine in every single wound site, you either cleave the fibers physically by cutting them, or you cleave fibers enzymatically by enzymes cutting the fibers in certain locations. And so fibers in wound sites are relaxed. Whereas, and you've seen that in the tissue, cells constantly pull on the fibers and keep them under tension. And so a very simple design of a polypeptide allows these adhesins to not just determine whether the protein is there or not, but to also probe the physical state of that protein. So the interesting part is fibronectin not just has bacterial binding sites, it is full with binding sites on every single domain against so binding sites for integrants, for main growth factors. It has cryptic binding sites that are only exposed if you denature the protein or perhaps stretch the protein. And we are exploring many of these binding sites in the moment, whether they are mechanoregulated or not. One way of doing that is you need to be able in cell culture to see whether or not the proteins are mechanically stretched. And the simplest way to doing that is to introduce optical probes, because optical microscopy is really the working horse in biosciences. And so what we do is we isolate fibronectin from human blood. We take advantage of 2 cryptic cysteines per fibronectin monomer that can be exposed and be labeled with acceptor dyes. Then we add a lot of donor dyes, and so roughly have a ratio of 7 donors to 4 acceptors per fibronectin dimer, and then measure FRET ratios. But since we have more than 1 donor acceptor pair, we say the FRET ratio changes on average if you stretch a protein, thereby change the average distances among all the donors and acceptors. And with such a technique, we can then indeed label fibronectin matrix in cell culture in 2-dimensional and 3-dimensional cell cultures. We can use fibronectin as a mechanical strain sensor, and we see in every field of view that many fibronectin conformations all coexist next to each other. But since we have many donors and acceptors, what does the color, false color, blue, for example, mean here? In order to calibrate our system a little better, we again pulled fibronectin fibers, and you see a relaxed fiber is red. We stretch a little more, the fiber turns green. We stretch a little more, the fiber turns blue. And so with our artificially pulled fibers, we can tune through the full range of conformations that you see in every field of view in cell culture. So indeed, with this FRET-labeled fibronectin, we have a mechanical strain sensor at hand. So the next question is, many of us are using antibodies. And we always assume that if the antibody doesn't bind, the protein is not there. That's the basis for most of our studies. But now imagine there are antibodies that have epitopes that can span several domains. And we have one antibody that binds in between these 2 domains. And we labeled now this antibody with a 3rd color so that we could do a FRET analysis of our stretched fibers. In this case, we stretched them vertically. You see the stretched fibers being greenish-blue, the relaxed fibers being red. If we now stain for the presence of the antibodies, you see that the antibody recognizes the relaxed state, but not the stretched state. So this is a very clear illustration that even antibodies, if they interact with mechanically strained systems, you need to know whether or not the epitope is mechanically strained, and cannot just conclude from the fact that you don't see an antibody that the protein might not be there. But it might be there, but it might be mechanically altered. So this field is very rapidly growing. It was made available by and accessible through all the advances that came from the nanotech community. What is really, really interesting as people are looking now into the many different proteins that only define this one type of solid adhesion sites, that many more, each protein has its own motif by which a mechanical stimulus is converted into a biochemical stimulus. So there's not one mechanosensor that is used by cells, but all the proteins that are part of force-bearing network seem to be utilized by cells in very intricate, interesting ways. So lessons to be learned are many different structural motifs. And the means by which force may alter protein function are therefore also quite diverse. You might expose cryptic binding sites that are otherwise may be buried in the inside of a protein by mechanical force. You might weaken or strengthen receptor-ligand interactions, and this is the second topic I would like to address. And you might open or close ion channels. Because all these protein networks, they can only work and perform well if the contacts between the proteins are sufficiently high that they hold on while the protein next to the contact is mechanically deformed. And so we need to look into the lifetime of bonds and how the lifetimes of bonds is mechanically altered by force. And what was very interesting certainly was as the biophysics community was exploring how bond lifetimes were changed by mechanical force, it was always seen that the bond lifetimes were shortened. But this is somehow not what you need when you want to use mechanical force to regulate function. And what was even more surprising is the amount by which a mechanical force can shorten the bond lifetime. So here, the biotin-streptavidine bond is one of the most commonly used bonds in biotechnology. It's a lifetime of a few days, but then if you just apply the force that is generated by a single motor protein, you can shorten the lifetime to 1 minute. So we use so often biotin-streptavidine interaction in microfluidic devices to build up our constructs and never think about lifetime shortening and under what conditions do we simply rupture off from the interfaces what we try to build. So how does nature really deal with these issues? And there seems to be a class of proteins and their ligands that evolved to form catch bonds. And catch bonds are bonds that get activated by mechanical force. And I would like to give you now brief insights into how these catch bonds work. Essentially, they are wonderful superglues that get stronger if you mechanically pull on them. And the first observation that such catch bonds really exist came from these videos. Where under low flow, we saw that a second bacterium, E. coli, and could you please also activate this video, where we see that the bacterium does not accumulate on a surface under low flow, but heavily accumulate on the surface at high flow conditions. So the question was first, is that an artifact? No. Our colleague Evgeny Zogorenko in microbiology had made many, many mutants. We could see point mutations in the adhesion that the bacteria use could change this flow-dependent adhesion to surfaces. And what E. coli has, and is shown here, it has long hair on its surface, and at the tip of each of these hair is a little adhesion molecule. And this adhesion molecule binds to mannoses. And what we could show then also was that if we coat a surface with antibodies, and an antibody will find an epitope on the outer surface of the adhesion, then we see the typical expected lifetime shortening as function of shear stress. However, if the mannose ring is allowed to slip into the binding pocket, we saw this flow-enhanced adhesion of bacteria to the surfaces. And the flow conditions under which we see enhanced adhesion are typical for physiological flow conditions. I just like to remind you that E. coli causes urinary tract infection, so it needed to optimize a mechanism to survive flow conditions and to populate niches that otherwise would be constantly cleaned. And you are probably also aware that we always get trained to wash our hands we rinse them under water. So here you see flow stimulates, activates E. coli to stronger bind to a surface. So how did that work? We worked on that question for 10 years now. What is the underpinning molecular mechanism? Did nature essentially evolve something that works like finger traps, where you can mechanically deform a certain pattern around a finger just by stretching it? And I would like to now show you the final scheme of how it works, and that was made possible only by solving then finally both structures. We postulated early on that there are two structures and that mechanical force convert from one structural state into another structural state. But how does that work? Here you see the long hair again of the fimbria. On top, you see the binding pocket for mannose, shown here schematically. But then you also see that the neighboring domains are in close contact to each other. And so what we find now is that this domain acts like a cofactor to regulate this binding pocket. And usually we're used to the existence of cofactors, but those were always soluble cofactors. Here it's a structural unit. And so if you now mechanically pull on this construct, you separate the two domains, these loops that are pressed towards each other and distorted a little in the structure, they get loosened up, and what we see is propagation of structural changes to the binding site and switching the binding site to a high binding state. And so it's essentially an allosteric effect, but it's a mechanically activated allosteric effect. So finally, there's certainly a lot that can be learned by looking at mechanics in living systems. And we now also need to ask the questions, how do we utilize all these insights for our own, to engineer new cells, to engineer new materials, to engineer new devices. And I would like to use this example to illustrate to you how far we are away from the man-made cell, the synthetic cell. So what you saw here in the video, and if we could just play that again, please, is a microtubule. And the microtubules are used in the cells to schlep cargo from one location to another. A cell is always operating very far from equilibrium. But then in the cell, you also need to stimulate where should the cargo be picked up and where do you want to deposit the cargo to make all our cellular functions possible. So what we would like to learn is how we can use molecular motors outside of a cell and then use them to transport cargo. We would like to learn how we can build motor-driven transport devices. These devices are powered by chemical fuel, and we would like to learn how we can utilize that then to sequentially assemble cargo of interest in a similar way to how nature uses it. And ideally, we would one day like to reach the state where we can build an assembly line for nano cargo at the nanoscale utilizing motors. And you might be aware that we use so far self-assembly approaches or top-down approaches of guiding, but we do not have good tools to engineer nano cargo and then to assemble it in the similar way to how we assemble our cars in factories. And as an illustration, let me give you this one. So you might recognize the border here of Switzerland. And over the last centuries, Switzerland has really optimized the ability to collect cargo or gold from the rest of the world and to bring it into Switzerland. And so we want to now build loading station for gold at the border of Switzerland and then transport, because we are all in need of some research funding, we would like to transport this money to ETH. And then this entire device should be not more than the diameter here of a human hair. And how can we realize that? How can we realize a cargo transport system at that length scale where you can thermodynamically stably deposit cargo, and then when a motor protein comes and collides with the cargo, it needs to pick up the cargo, transport it, and then you need to engineer another chemistry to deposit the cargo on demand. And this is where mechanics comes in very powerfully again. There was very beautiful work done by Hermann Gaub's group showing that you can use oligonucleotides, and the number of base pairs certainly determines the thermodynamical stability. But like having a zipper, it makes a huge difference whether you open it bond by bond by bond by bond, or whether you would try to open it this way. And so you immediately see that while the thermodynamic stability is just given by the number of hydrogen bonds that is formed, the mechanical stability can be tuned by the geometrical arrangement in which you apply force to open these clusters of hydrogen bonds. And we utilize that in our design. So now you see we deposited 40 nanometer gold cargo on the border of Switzerland in the zipper design, and then made unloading stations in the shear design. And now I would like to show you just briefly that this entire system also works. And here is a microtubule that moved across the surface. It was driven by kinesin. So what we had done here, we deposited the cargo, we bound kinesin in an inverted geometry on the surface. In the presence of ATP, it could bind to a microtubule, which we use as our molecular shuttle. It can push this microtubule forward. Then the microtubule, which is itself only 30 nanometers in diameter, has to collide with a few of these nanodots on the surface. And during the collision, enough force needs to be generated to pull off the cargo from the border here and transport it over to the unloading station. And if I could perhaps just show you this video again. The design works. And so what is very, very important, and the take-home message here is that when designing complex devices in the moment with supramolecular assemblies, to think about, first of all the biochemical aspects, all the chemical aspects, but then also to ask, okay, if I now apply force to a protein, to a subunit, to a bead, what is happening with my bonds that are supposed to thermodynamically stabilize the interactions? So we try to utilize a lot of the new technology that is available, and certainly chemists and physicists and engineers bring a lot of new technology that traditionally were not used in the bioscience community. And we have enormous potential to make contributions, to ask questions that are different from questions that a person trained in biosciences would usually ask. So we like to understand at all the different length scales how proteins act as mechanical chemical signaling switches, how a cell sense and then respond to mechanical stimuli, how their ability to respond to mechanics impacts finally tissue growth and regeneration, and how we can use force to help in regenerative processes. And finally, to also understand why exercise and food keeps us healthy. And we would like to utilize basic science principles to then derive a new set of drugs that might target non-equilibrium structures, which has so far not been done. We would like to, this information can be used to better design interfaces that can control cell functions. And why is this entire topic so important to be addressed right now? You might recognize there are many, many diseases for which we so far have no cures. So we treat the symptoms of the diseases, but do not really know how to revert the symptoms per se. And these diseases, first of all, I should not talk it by disease, but even the question, how and why does exercise keep us healthy is unsolved. We know it's not just training by our muscle system. It's not just training our cardiovascular system, but Exercise seems to downregulate inflammatory responses, and the mechanisms how this is happening are unknown. Many diseases exist that are associated with mechanical over or understimulation, just to name the two major ones, cardiovascular diseases and osteoporosis, and this is where we have nothing available to revert the progression of the disease. Why do cancer cells have an altered mechanoresponse system, and how does that impact malignancy is unclear. Why stem cells and how they can be guided by physical features in their environment is unclear. So we need to understand at the molecular level how forces regulate function. It's absolutely essential for many, many fields to come and to progress. And since I was also asked yesterday about smart materials, what are smart materials? And I thought what was on the list there were maybe just 2 or 3 stars out of an entire sky that you might not necessarily see as a city person, but then in the countryside. So there are many, many more stars that can be utilized to make smart materials. And I think the next wave of innovations in the biomedical field will come not only from us using nanoparticles in nanomedicine, but the next big series of innovations will come from us studying, using nanotools, nanoanalytical tools, how the nanoscale biological machinery is working, and then to convert that knowledge into innovation and new discoveries. And with that, I would like to thank, this work is certainly only possible with having wonderful colleagues in all kinds of disciplines and all around the world. We are part of a nanomedical center where we get a lot of good stimulation and interactions. And then I would like to thank all the people that have done the work and other collaborators, and very much you for your attention. Thank you.